For the third and final time this semester, I'll introduce myself. I'm Greg Tilka from Plant Pathology, and for the second time in 8 days, it's my pleasure to introduce seminar speaker. Today we have Dr. Nathan Schroeder from University of Illinois, and if you've heard me introduce people in the past, I like to pull out tidbits of their upbringing and so forth. So he has an Iowa connection. Seems like anything that happens in the world, the news finds an Iowa connection. So he has an Iowa connection we'll get to. But he was born in Georgia and very early in life moved to the Syracuse area of New York where he grew up. He got a bachelor's degree at Earlham College in Indiana, right? And a PhD at the University of Wisconsin with Ann McGoodwin. His Iowa connection is his dad grew up over near Lorville, which is over near Carroll, Iowa, and still owns land there. And his uncle, your dad's brother, presumably, brother-in-law, still farms just south of Fort Dodge here in Iowa. So he said he'll probably inherit some Iowa farmland at some point.
Yeah, once my dad kicks the bucket.
Yeah, it's always good to have goals.
Yeah.
After he got his PhD, he did a postdoc at Rutgers University working on C. elegans, and he's been at the University of Illinois since 2013. He's an assistant professor, and his lab works on the development and neurobiology of nematodes. And without further ado, I'll turn it over to Nathan.
Okay, so why is SCN fat and lazy, and other important nematode questions? So I was getting this talk ready on a day that I had skipped lunch, and so I was starting to get hungry. And so I had a food theme because everyone likes to think about food when they think about worms. So what is a nematode? Why should you care? Sort of the introduction. And then I'm going to talk a little bit, some pieces of data from some published work. And then the main course is going to be stuff that we've been working on for about the past year and a half that's unpublished. So it will be somewhat incomplete, but I would love suggestions or comments. And then what I think most people would agree is the best part of any meal is the dessert questions. If I run out of time, we don't have to be part of the Clean Plate Club, so we want to save room for dessert, so I may cut it short if need be. Okay, what is a nematode? Why should you care? So why should you care? Well, if nothing else, nematodes are all around us. A few of these statistics are statistics that I don't necessarily vouch for, but I put the reference on there so you can be sure that I'm not just making it up. So nematodes are everywhere. Nematodes can serve as models for higher organisms. So the example is C. elegans, which has been a genetic model organism for the past 40 years or so. And then, of course, topic of interest for many of those in this room, parasitic nematodes. So there's both animal parasitic nematodes as well as plant parasitic nematodes that can cause extensive damage. So what is a nematode? Here is one textbook definition, but the main thing, and here's a few headshots of different nematodes, but the main thing, especially talking to a plant pathology audience, that makes nematodes somewhat unique for most plant pathogens is that they are animals. Okay, so they're not bacteria, fungi, or viruses. Because that they are animals, they also have a lot of the features that animals have. So they have muscles, they have neurons, they have an epidermis that secretes a cuticle. So if we took a typical nematode and cut it right in the middle, we would see quadrants of muscle tissue. We would see a bunch of neurons in a big bundle down here on the ventral side. We'd see another little bundle on the dorsal side. We would see a thin layer of epidermis around that whole structure that secretes the outer cuticle. Just to get us sort of on the framework of what nematodes are again, so nematodes are their own phylum. Based on current molecular data, we divide the phyla into 12 different clades. So C. elegans is here in clade 9. Most of the plant parasitic nematodes, and in fact all the ones I'll talk about are here in clade 12. But the aside from this, the point to get across is there's a lot of different types of nematodes out there. Most of them are what we call free-living. They feed on bacteria or fungi. The other thing to note is that parasitism, whether it's on plants, on insects, or so-called higher organisms, this has evolved multiple times. Okay, so plant parasitism has evolved probably at least 4 times. So there's a fair amount of diversity out there. Okay, so as Greg mentioned, we're really interested in sort of the development of nematodes. How do they develop? How do you get a nematode? You start off from a single cell egg. It has to form that structure. How does it get there? So to give a sampling of data from my lab and what I've done, so as Greg mentioned, I started my PhD at Wisconsin. I worked on plant parasitic nematodes there. I then went to a C. elegans lab. So C. elegans is a bacterial-feeding nematode. It has a lot of really great attributes. It has a genetic model system, and we still, in my lab, do about, I would say about 40% of our work is on C. elegans. So just to give you an idea and to sort of introduce you, for those of you who aren't familiar with nematodes. So nematodes, including C. elegans, start off as an egg. They develop through 4 larval or juvenile stages. So I'll alternate between L1 to L4 and J1 to J4. They mean the same thing. It's just different communities use different terms. So my research as a postdoc was actually focused on a neat trick that C. elegans does that not all nematodes do. So C. elegans, if we grow it on a petri dish with bacteria, it will hatch into an L1. It'll then feed and develop through these stages into the adult. If it hatches as an L1 and it senses that the environmental conditions aren't good, it can make an alternative developmental choice and go into this stage called a dower, which is analogous to some of the infective stages of parasitic nematodes. So coming from a parasitic nematode background, I was very interested in this particular stage and behavioral changes that the dowers had. So that's been sort of a focus that I had. So in particular, I focused on a set of 6 neurons. So one of the great tools that was built in early on in the C. elegans culture was that a set of researchers basically went through the adult stage of C. elegans. They fixed the animals and they did serial section EM. So they basically took that animal, it's about 1 millimeter in length, and they did 70 nanometer sections over and over and over again. And then they reconstructed from basically nose to tail the entire nervous system. So every single neuron in the adult stage is known, and all of the connections between neurons are known. Okay? So I was looking at a set of neurons in the head called the IL-2 neurons. And for those of you who remember your high school biology, so neurons will have processes, dendrites, and axons that receive or send signals. So these neurons are in the head. The tip of them are right at the tip of the nose. And throughout this talk, I'm going to try and, most of the pictures, I'm going to have the orientation with anterior to the left, just in case you kind of get confused about what's what. And so this was the structure of these IL-2 neurons during non-dower stages. And this had been known for 30 years. And so I guess my big discovery was finding that during dower, what happens is that instead of looking like this, the neurons look like this. So they undergo all this extensive arborization of the dendrites, such that they increase about 3 to 4-fold in length. Okay? And we've actually done some work that's showing that these have behavioral effects that are specific for dower-specific behaviors. So after dower, if we take those dowers when they're stressed out and we put them back onto plates with lots of food, they'll come out of dower, and basically all of these branches will go away. And you'll just have these tiny little remnant branches. And that'll happen within about a period of 12 hours. So we've got part of the lab that's working on that. But during my postdoc, I was still very interested in plant parasitic nematodes, so I was kind of the oddball in the lab. I went to my garden, I